Hello and welcome dear students. Today we are going to discuss about the topic from Mendel to molecules. The main objectives of this module are to know about the history of the science of genetics, to know pre-Mendelian ideas on heredity, to know about the background for modern genetics, to understand the fertilization and gestation of modern genetics, to understand the emergence of molecular genetics, and finally, to know the scope of genetics. Dear students, let's start with the introduction first. Genetics as a scientific discipline started from the work of Gregor Mendel in middle of the 19th century. Mendel suspected that traits are inherited as discrete units, even though he knew nothing of the physical or chemical nature of the genes at that time. His factors or units became the basis for the development of the present understanding of heredity. All the current research in genetics can be traced back to Mendel's discovery of the laws of inheritance. In 1905, William Bateson introduced the term genetics, who was one of the strong advocates of Mendel's work and became a champion of Mendel's principles of inheritance. Dear students, let's know about the history of science of genetics. The scientific evidence for patterns of genetic inheritance was started from Mendel's work. History indicates that humankind must have been interested in heredity long before the dawn of civilization. This is because of the curiosity based on human family resemblances such as similarity in body structure, voice, gait, and gestures. Early nomads used the quality of the animals to breed and domesticate, indicating selective breeding. The first human settlement was practiced farming with selected crop plants with favorable qualities. Besides, ancient tomb paintings show a racehorse breeding pedigree, indicating the inheritance of several distinct physical traits in the horses. Moreover, the first recorded information on heredity was found during the time of the ancient Greeks. Some of their ideas are still considered relevant today. Dear students, Hippocrates, who lived during 460 to 375 BCE, was known as the father of medicine. He believed in the inheritance of acquired traits. To propagate this, he devised the hypothesis of pangenesis. He put forth that all organs of the body of a parent gave off invisible seeds. These were like miniaturized building components that were transmitted during sexual intercourse, resembling themselves in the mother's womb to form a baby. He also proposed that materials from all parts of the body concentrated in male semen. This was then developed into a human in the womb of the mother. For example, the massive biceps of an Olympic weightlifter were present as many bicep parts in the semen. Therefore, his children would also have big biceps. Thus, he believed in the inheritance of acquired characteristics. Later, Aristotle criticized his theory. He objected that mutilated and physically disabled people could have normal children, as well as more subtle people can transmit characteristics that do not show at conception but develop at a later age. Aristotle, who lived in 384 to 322 BCE, pointed out the importance of blood in heredity. He believed that the blood material was for building all parts of the adult body. He proposed that blood was the basis for passing on the generative power to the next generation. He also put forth that the male semen was purified blood. The woman's menstrual blood was equivalent to the semen of males. Therefore, the male and female contributions united in the woman's womb to produce a baby. The blood had some type of hereditary essences. He also believed that the baby would develop under the influence of these essences rather than being built from the essences themselves. He rejected the bricks and mortar model of hereditary transmission and proposed a blueprint model. For both Hippocrates and Aristotle, and also all other scholars through the late 19th century, believed that the inheritance of acquired traits or characters was a well established fact that any theory of heredity had to explain. In addition to this, individual species were taken to have a fixed essence. Those inherited changes were merely superficial. Later, the Athenian philosopher Epicurus observed families and proposed the contribution of both males and females of hereditary characters as sperm atoms. He noticed the dominant and recessive types of inheritance and described the segregation and independent assortment of sperm atoms. In the Charaka Samhita of 300 BCE, ancient Indian medical writers noted the characteristics of the child determined by the four factors. They are those from the mother's reproductive material, the father's sperm, the diet of the pregnant mother, and accompanying the soul which enters the fetus. Each of these was the four parts, creating 16 factors, of which the karma of the parents and the soul determined attributes predominantly and thereby gave the child its characteristics. Dear students, let's now understand about the background for modern genetics. The 4 L's. A mathematical version of pangenesis was developed as the biometrical school of heredity by Darwin's cousin Francis Galton. In between Aristotle and Mendel's time, a new idea was introduced on the nature of heredity in the 17th and 18th centuries as the concept of preformation. Scientists, by using the newly developed microscopes, imagined that they could see miniature replicants of human beings inside sperm heads. Dear students, let's now understand how the 4 significant L's contributed to the development of the science of genetics. The first, von Leeuwenhoek, between 1632 to 1723. The major contribution of von Leeuwenhoek was the significant improvements to the microscope, which made him observe what he termed animalcules, today as microbes or microorganisms. Several scientists considered von Leeuwenhoek as the father of microbiology because he was the first to report on single-cell organisms. Scientists of the day greeted his findings with suspicion and resistance. Only a team of impartial researchers replicated his observations that microbes were accepted as a real phenomena. Therefore, his significant contribution was to initiate a whole field of science that subsequently led to the identification of chromosomes and the development of cell theory. Now the second one, that is Linnaeus. He was a Swedish physician and biologist, contributed to the classification of biological entities, a scientific document of great popularity at that time. He published his first edition of the book Systema Naturae in 1735. In this book, he provided the seeds for modern biological classification. Therefore, his significant contribution was the sets of rules for classification which gradually became universally accepted. The main salient features of the Linnaean system are: it is binomial system where two names call an organism its genus and species using Latin but sometimes Greek roots. For example, the scientific name for man is Homo sapiens. The second, the binomial system was mainly based on external morphology. Third, the system was hierarchical, that is, he believed in the biblical account of creation. Thus, he viewed species as being created and then remaining fixed and immutable. Now, Lamarck, who lived between 1744 to 1829, Lamarck was considered as the first modern evolutionist. He was the first to develop a comprehensive theory on the process of evolution. During that time, Erasmus Darwin predicted that a giraffe and a camel are the manifestations of one living filament. The idea was taken further by Lamarck and proposed that camels and giraffes have a common ancestor. However, they are differentiated by their adaptations to different environments. In lush environments, giraffes in the beginning ate the lower leaves of a tree and later stretched their necks to reach the higher leaves. This feature was then transmitted to its offspring, and after many generations, many animals with very long necks and members of this species, that's now the giraffe. Linnaeus' hierarchical classification implies that certain species are closely related while others are distantly associated. Lamarck got right with the concept of change as a function of heritable transmission and also adaptation to the environment. This was the mistake he made, and for that, his idea of the inheritance of acquired characteristics was not accepted. Now, let us discuss the contributions of the Lyell. Charles Lyell, a Scottish Turner geologist, Lyell's contribution to pre-genetic thought are much less attributable to him as an individual than they are to his overall field. Lyell's significant contribution was that he compiled and systematized the idea that became the origin of modern geology. His opinion on the origins of the earth and biological species were considered in Genesis, the first book of the Bible. Around 1600, Bishop James Ussher proposed the estimates of the geologies in Genesis to conclude that creation occurred on October 23, 4004 BCE. Dear students, let's talk about the fertilization and gestation of modern genetics. It took over 50 years for genetics to emerge as a unified field. If the time of birth of genetics is considered, then the fertilization and gestation began with 3 disparate developments during the 19th century. Of these, 2 are associated with individual Charles Darwin and Gregor Mendel. The third is new cell biology. Let's now discuss the contributions of the Charles Darwin. Charles Darwin was selected for expedition since his zeal for naturalism as a naturalist on HMS Beagle, the ship commissioned to chart the waters along South America's coast. During 5 years' voyage, he systematically noted his observations to Cambridge. His travels exposed him to fossil collections of extinct species that showed striking similarities with existing species. During this time, he was impressed with Thomas Malthus' essay on the principle of population. In the next 2 years, he connected the dots of his ideas and observations and developed the incipient stage of his natural selection theory. British naturalist Alfred Russell first postulated the theory of evolution by natural selection. The principle of natural selection should be termed Darwin and Wells's theory of natural selection in 1859. Gregor Mendel. Gregor John Mendel, an Austrian monk, performed rigorous experiments on inheritance and genetics of sweet pea plant. He developed the unit of heredity in 1865 as a particle that does not change and is passed on to further to offspring. His work is the basis of understanding the principles of genetics even today. Consequently, Gregor Mendel is known as the father of genetics. Dear students, let's talk about the birth of cell biology. In 1665, the Englishman Robert Hooke used the term cell to describe the compartments of cork and other plants because they remind him of cellula. The role of cells was a hotly debated topic until the development of cell theory by German botanist Matthias Schleiden and German physiologist Theodor Schwann in 1838 and 1839 respectively. This theory held that all life is composed of cells and that only origin of cells was from other cells. A significant advance was the detection of chromosomes and the unraveling of cell division. They were first reported by Carl Wilhelm von Nageli in plants and Edward von Beneden in animals. Voltaire later named them as chromosomes in 1888. Dear students, let's discuss about the rediscovery of Mendel's work. It was in 1900 when three people found Mendel's theory. They are Dutch botanist Hugo de Vries, the Austrian Erich von Teschermark, and the German Karl Correns, who were working on the hybridization technique independently. Hugo de Vries wondered what the nature of germplasm might be, and in particular, he wondered whether or not germplasm was mixed like paint or whether the information was carried in discrete packets that remained unbroken. In the 1890s, he was conducting breeding experiments with a variety of plant species, and in 1897, he published a paper on his results that stated that each inherited trait was governed by two discrete particles of information, one from each parent, and that these particles were passed along intact to the next generation. In 1900, he was preparing another paper on his further results when he was shown a copy of Mendel's 1866 paper by a friend who thought it might be relevant to de Vries' work. He went ahead and published his 1900 paper without mentioning Mendel's priority. Later that same year, another botanist, Karl Correns, who had been conducting hybridization experiments with maize and peas, was searching the literature for related experiments prior to publishing his own results when he came across Mendel's paper, which had results similar to his own. At the same time, another scientist, Erich von Teschermark, was experimenting with pea breeding and producing results like Mendel's. He too discovered Mendel's paper while searching the literature for relevant work. In a subsequent paper, de Vries praised Mendel and acknowledged that he had only extended his earlier work. So this was the rediscovery of Mendel's actual experiments. Dear students, let's understand about the emergence of molecular genetics. After the rediscovery of Mendel's work, there was a feud between William Bateson and Pearson over the hereditary mechanism followed by Ronald Fisher in his work, The Correlation Between Relatives on the Supposition of Mendelian Inheritance. In 1910, Thomas Hunt Morgan showed that genes reside on specific chromosomes while working with Drosophila. He later showed that genes occupy specific locations on the chromosome. With this knowledge, Alfred Stuyvesant, a member of Morgan's famous fly room using Drosophila melanogaster or fruit fly, provided the first chromosomal map of any biological organism. In 1928, Frederick Griffith showed that genes could be transferred in what is known as Griffith's experiment, injections into a mouse of a deadly strain of bacteria that had been heat killed transferred genetic information to a safe strain of the same bacteria, killing the mouse. A series of subsequent discoveries led to the realization decades later that the genetic material is made of DNA, that is deoxyribonucleic acid, and not, as was widely believed until then, of proteins. In 1941, George Wells Beadle and Edward Lavery Totem showed that mutations in genes caused errors in specific steps of metabolic pathways. This showed that specific gene code for specific proteins, leading to the one gene, one enzyme hypothesis. Oswald Avery, Collins Munro, MacLeod, and MacLean McCarthy, three scientists, showed in 1944 that DNA holds the gene's information. In 1952, Rosalind Franklin and Raymond Gosling produced a striking clear X-ray diffraction pattern indicating a helical form of DNA. Using these X-rays and information already known about the chemistry of DNA, James D. Watson and Francis Crick demonstrated the molecular structure of DNA in 1953, which is considered to the remarkable discovery till date. Together, these discoveries established the central dogma of molecular biology, which states that proteins are translated from RNA, which is transcribed by DNA. This central dogma has since been shown to have exceptions, such as reverse transcription in retroviruses. In 1972, Walter Fiers and his team at the University of Ghent were the first to determine the sequence of a gene, the gene for bacteriophage MS2 coat protein. The successful sequencing of many organisms' genomes has complicated the molecular definition of the gene. In particular, genes do not always sit side by side on DNA like discrete beads. Instead, regions of the DNA producing distinct proteins may overlap, so that the idea emerges that genes are one long continuum. It was first hypothesized in 1986 by Walter Gilbert that neither DNA nor protein would be required in such a primitive system as that of a very early stage of the Earth if RNA could serve both as a catalyst and as a genetic information storage processor. The modern study of genetics at the level of DNA is known as molecular genetics, and the synthesis of molecular genetics with traditional Darwinian evolution is known as the modern evolutionary synthesis. Dear students, let's know about the scope of genetics. Geneticists are employed in the various brewing industry to improve the strains of yeast to produce alcohol. The pharmaceutical industry has developed different strains of molds, bacteria, and other microorganisms high in antibiotic yield. Penicillin and cyclosporin from fungi and streptomycin and ampicillin from bacteria are some examples. Agriculture and animal husbandry apply various genetic techniques to improve plants and animals. Selective breeding and transgenic modifications using recombinant DNA techniques are routinely used. Livestock breeders use artificial insemination to propagate the bull's genes. Several types of mammals are cloned to explore certain desirable products. With knowledge of genetics, several highly specific diagnostic kits have been developed to diagnose different types of human diseases. This can be detected in embryos as young as 12 weeks by using amniocentesis or chronic villus sampling. Stem cells from embryonic tissues are for early treatment of specific genetic abnormalities. Gene therapy is the modification of defective genotypes by adding functional genes made through a technique called recombinant DNA technology. With family history, genetic information is used to prevent, diagnose, treat, and manage many complex diseases. Currently, we are in the era of personalized or precision genetic genomic medicine. To conclude, genetics is a branch of biology concerned with the study of genes, genetic variation, and heredity in organisms. Every individual inherits genes from both their biological parents, that is, father and mother, and these genes in turn also express specific traits. It mainly deals with how traits are passed from parents to their offsprings. The 100 years of vast knowledge on genetics can be used in any field and in any organism to understand their biology, explore their significance, and treat any disease. Dear students, let's now conclude the lecture. Genetics is a branch of biology which deals with the study of genes and their inheritance from parents to offsprings or from one generation to the other. It was started by Mendel, father of genetics, who worked on pea plants, providing 3 laws on heredity, even though Mendel did not know anything about the genes at that time. Genetics has always been an enigma for humanity, and the process of inheritance has piqued our interest since time immemorial. This was based on human family resemblance, such as similarity in body structure, voice, gait, and gestures. Hippocrates, who is the father of medicine, strongly believed in the inheritance of acquired characters and proposed the theory of pangenesis. Later, Aristotle suggested the importance of blood in the human body. He put forth the idea that male semen purified blood and female menstrual blood united to produce a new organism. Subsequently came four philosophers commonly called 4Ls who laid a strong foundation for the development of modern genetics and evolution. Antony van Leeuwenhoek, the father of microbiology, invented the microscope that made him see microscopic organisms. His contributions to the field led to the development of cell theory. Carl Linnaeus then came with the idea for modern biological and bionomical classification, which is universally accepted. Lamarck proposed the idea of evolution. The theory is called Lamarckism. Charles Darwin, who went on an expedition, proposed the theory of natural selection and evolution. The birth of cell biology took place when Robert Hooke discovered the cell. Sheldon and Shivon proposed the cell theory. The research in the field led to the development of various branches in genetics having a wide variety of scopes. Dear students, it was all about today's lecture regarding from Mendels to molecules. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.